the two contractile proteins in the muscles are actin and myosin. So actin, the polymers of actin are going to kind of wind together and make up these thin filaments, which are kind of more continuous than the other filaments. And then the thick filaments are a little bit more staggered, and they're made of myosin. And so the tails of the myosin filaments adhere to one another, and this is going to actually keep the thick filament as like a actual thick filament with many myosin filaments together. And so the head of the myosin filaments are going to kind of stick out, and they kind of look like these little golf clubs here. And these little heads are actually going to bind to ATP, which is actually going to allow the muscle to contract. And so this entire thing, this whole long thing of the diseases and the thin filament and the thick filament, is actually called the sarcomere, which is kind of the smallest unit of contraction in the muscle. And this is also why we see like striations under a light microscope, is because the edges of the sarcomeres line up with such striations. So then, in addition to the contractile proteins, there are also regulatory and structural proteins. And so the regulatory proteins are tropomyosin and troponin. So basically, what they're going to do is basically allow the muscle to contract, and they're going to turn it off, contraction on and off. And they're going to do this by blocking off the myosin head, because if ATP can't bind to the myosin head, the muscle can't contract. So they can kind of control whether or not the myosin heads are exposed, and thus they are going to allow the muscle to contract or be relaxed. And then there's also these structural proteins which aren't in this picture, but some of them are nebulin, dystrophin, another one is tiny, and basically what they're going to be doing is they're going to provide proper alignment and allow the muscles to extend and be elastic, which is important if you are a muscle and need to do those things. So in order for a muscle to actually contract, you need an action potential, which comes from the nervous system. So this action potential is going to come to the synaptic terminals of the motor neuron, and it's going to trigger the release of acetylcholine, which is supposed to be these red dots over here. And the acetylcholine is going to come and it's going to bind to these receptors on the membrane, or the plasma membrane of the actual muscle and muscle cell. And then the action potential is going to travel along the plasma membrane. And it's going to keep going along here until it hits the T tubules, or the transverse tubules, which basically are infoldings of the plasma membrane. And then the action potential is going to travel along here, and it's going to hit the sarcoplasmic reticulum. And the sarcoplasmic reticulum it's just like a special endoplasmic reticulum, but it's full of calcium ions. And so when it's hit with the action potential, it's going to change shape. And that's going to open these calcium channels. And then the calcium, which is really concentrated in here, is going to diffuse out into the cytosol. And so then these calcium ions are going to bind to the troponin complex over here, which is supposed to be this little purple thing. And in the actin filaments, this is an active filament. And so the calcium ions are going to kind of start they're going to initiate the thick and thin filaments kind of sliding, and this is going to expose the myosin heads so ATP can bind to them. And then I need another picture to actually explain how muscles contract. Okay, so after the myosin has been exposed, then the ATP can actually bind. So ATP is going to bind to the myosin head, except the myosin head is initially in its low energy configuration. And ATP is going to hydrolyze, and it's going to become ADP, adenosine diphosphate, and an inorganic phosphate. And then so it's going to phosphorylate the myosin head. And then the myosin head is going to gain energy. It's going to become in its high energy configuration. It's going to actually change shape. And then so when it changes shape with the ADP and the inorganic phosphate present, it's going to bind to the actin. And this is going to make what's called a cross bridge. And so then this cross bridge is like the myosin head is actually binding to the actin filament. And so then when the ADP and the inorganic phosphate are released, it's going to make this like power stroke and like push the actin filament towards the center of the sarcomere. And so many of these filaments doing this together is actually going to contract the sarcomere, and like spend many sarcomeres doing this, like going to contract the myofibrils, and then many myofibrils doing this will actually contract the muscle. And so then you can have this cycle keep going to keep the muscle contracting. So you can have ATP bind to the low energy configuration myosin head and keep this whole cycle going, which will keep the muscles contracting. And then to explain how a muscle relaxes, is to go back to this picture. So when the action potential that initially triggered the muscle to contract is over, then the calcium ions are going to start being pumped back into the sarcoplasmic reticulum, and it's going to use active transport to do this. So it's going to have to use ATP to transport the calcium ions across their concentration gradient, so in the opposite direction of the concentration gradient. And then, so after you get a low enough concentration of these calcium ions in the cytosol, it's actually going to trigger the regulatory proteins to go back to their initial state. And that's going to block off the myosin head so they can't bind to ATP anymore. And then the muscle is going to relax and not contract anymore. And so this is just kind of like a more macroscopic view of what's going on in the sarcomere. And so you can see when the muscle's relaxed, the fibers are kind of elongated. They're less thick, they're not, the actual fibers are all the same length. And then when the muscle contracts, the fibers kind of overlap and get really compact. And that's what is happening. And then many sarcomeres doing this is actually going to contract the muscle.